And in terms of misconceptions, I think that probably the biggest one is that V beta alone is sufficient. You know, people are like, oh, I can just look at V beta and I can see whatever I want. It's like, no, there's actually a lot more going on in the immune repertoire.
Welcome to the Repertoire Room. My name is Sander, co-founder and CEO at ImmuneWatch, an immunoinformatics company specialized in T-cell receptor sequencing analysis. And a couple of years ago, I got completely obsessed with T-cell repertoires. And I know that I am not the only one. So in this podcast, I want to have insightful conversations with people that share this passion and discuss TCR sequencing technologies and analysis in general. And in this very first episode, I'm actually honored to be joined by someone I had the pleasure of working closely with in the last year, the great Jonathan Doose, Executive Director of Business Development at iRepertoire. Welcome, Jonathan.
Hey, thank you for having me, Sanders. It's fantastic to be here.
Yes, great to hear. The first episode, so let's just dive in this, Jonathan. I believe that not all careers are the same, so I am wondering, what's your origin story like? How did you actually end up in this whole repertoire field?
Yeah, it's been an interesting career path for me. So I originally studied to be a medical doctor. So I worked on a bachelor's in biochemistry. I did a minor degree in neuroscience. I figured, oh, this is going to be, great prep for medical school. And then I shadowed a bunch of physicians. And then I thought, oh, I don't really want to be a physician. And so I kind of followed the path of least resistance. I was a lab manager in a breast cancer research lab, and I taught in the Learning Center. This was at UC Riverside a long time ago. So I taught organic chemistry. And anyway, the PI of that lab mentioned that there was another predoctoral fellowship. And so I applied for it and got it. And so I basically got into grad school because it was an open path for me at the time. And I was like, okay, this will be interesting. And then kind of reoriented my career towards more translational applications. So I moved from that breast cancer research lab, which really like the most exciting assay they did was cell migration on a transwell filter. So not super exciting. And I joined a neuroinflammation lab that was studying Alzheimer's, Parkinson's, and multiple sclerosis. And so I worked on a PhD for about 4.5 years in that lab. And I started my family young. So I got married my junior year of my undergrad, and my daughter showed up second year in grad school. And I watched my bank account get smaller and smaller every year. And I thought, oh man, I need to get out of here and earn some money. I gotta support a family. So I left grad school with a master's degree and did some soul searching and thought, well, what am I going to do? I was kind of done with lab work because I had just done so much of it. I mean, I made a transgenic animal. I did a lot of lentiviral overexpression and knockdown. I was studying one particular molecule that was upregulated anytime there was wound healing and protection happening in the brain. I was studying microglia in the brain, so not really T-cell per se. It was more on the myeloid side of immunology. But yeah, I ended up joining Clonetech Takara. I used a lot of their products in grad school, so I became their Los Angeles territory manager for a number of years, and the traffic in Los Angeles just about killed me. So I moved the family to North Carolina, still with Clonetech Takara. Did that for, I think, another 1.5 years, and then I joined a small qPCR company called Quanta Biosciences that had been acquired by QIAGEN. And that was interesting because it was a really large territory, like a lot of travel. I was covering, I think, 15 states, 70% overnight travel. It was just a lot of travel. And I was working with the VWR reps, so the life science specialists and the account managers. And that was really rewarding because there was a lot of teaching and coaching happening there. A lot of the VWR reps, I mean, it's variable. You find some that are good hunters, but most of them, I would consider them to be more like farmers. They just kind of cultivate their existing relationships. And so showing them how to map out their business and identify people to go talk to about qPCR. And then QIAGEN acquired Enzymatics, and they were kind of rolling Enzymatics and Tiangen and SA Bio and Quanta Biosciences into this new business unit called Custom by QIAGEN. And I had moved from a sales role into a marketing role at the request of a couple of the higher ups at QIAGEN to help rebrand Quanta Biosciences as Quanta Bio. And they wanted me to move up to Beverly, Massachusetts. And I'm like, well, I've already done one big move. I don't know that the family is going to be very wild about that. But my wife's family lives in Colorado, and there was a small company that was spun out of Enzymatics called Archer DX, and they were hiring product managers. Well, never done product management, but it seems like it would be a very interesting role. It would probably help me round out my commercial understanding of business development. So we did another cross-country moved to Colorado, where I am now. So I did that with Archer DX for a while. I was their product manager for solid tumors and cell-free DNA and inherited Mendelian disorders. That was a really stressful job. Just in product management, like so much is expected of you, yet nobody has to do anything that you say or ask. And so you spend a lot of time really trying to convince people, like, you know, we really need to do this. And then creating a lot of the quality documents, because Archer was pursuing ISO 13485, which is a really rigorous quality system. And we had a quality team of 3 people. And so the product managers ended up creating a lot of the quality documentation, which that's fun when you're dyslexic. Try to create an error-free document. You know, it's just very stressful. But yeah, they did a riff. And so I left Archer and then went to Agilent for a bit, their diagnostics and genomics division. And then I went back to Archer in a sales role for a little bit. And then they got acquired by Invitae, which was an interesting acquisition. It didn't really make a lot of sense to me. And I saw some of the things that looked like they could lead to another riff. And I was like, yeah, this doesn't seem right to me. So I ended up joining Olink Proteomics and did that in the Midwest for a while. Did really well. Made their top 10 salespeople in that first year, brought the Mayo Clinic online. And then, yeah, the gentleman who I reported to at Archer, Jason Amsbaugh, he had started a marketing firm, Samba Scientific, and one of his clients, iRepertoire, was looking to build their sales team, and they were ready to make their first sales hire. And so my name was recommended for that. It was never a job that was posted, never really applied. They just, hey, it was more of a networking thing. And so I started discussions with them and thought, yeah, this actually aligns really nicely with my background. And, they a small company, they need someone who can wear a lot of different hats. And so I thought, yeah, let's do this. So I left a very well-paying job at Olink and joined little startup company in Huntsville, Alabama. And yeah, so I've been there for almost 2.5 years now. Executive director of business development. So I wear lots of hats. Back in grad school, doing a lot of gene engineering. Ever since moving into the commercial space, I've been doing a lot of sales process engineering. And so that's really what I've been doing at iRepertoire, is just building a lot of the fundamental structure of how do we approach customers, how do we engage, how do we build trust, how do we scope projects, how do we create all the different contracting templates that we need in order to onboard clients. And it's been very rewarding. The business is growing very fast, and it's creating a bit of a challenge because we need to grow. We need to hire more people because we have a lot of business that we're carrying right now from a small team, and AI is helpful because it can kind of be like automation, a force multiplier. You can get more done with fewer people.
Yeah. Okay. Cool. Indeed. Yeah. So coming from the lab, from an MD career, maybe looking to the lab, sales roles, product manager, and now executive business development. Yeah, interesting. But yeah, iRepertoire indeed focuses on this whole T cell, B cell repertoires. Mm-hmm. But I guess then Archer also previously has these kind of technologies available. So it's not, iRepertoire was not your first touch point with T cell receptor analysis, right?
No, not really. I mean, I did not oversee the Immuniverse product at Archer DX. So Darius Fougere, a good friend of mine, we shared an office. And so, yeah, we had many conversations as he was working on the projects that he was developing, and then me for mine. So, yeah, and because I was like the token salesperson on the product management team, I would do a lot of co-travel in the field and work with the sales rep. So we had a number of conversations that moved into the repertoire space. But Yeah, it's definitely with iRepertoire. My focus on repertoire
Okay.
analysis is now much more
Yeah.
all-encompassing.
Okay, so you're also as newly excited as I am about this whole sequencing space, because it's 2.5 years now. Yeah, for me, it's about 4 years ago that I transitioned from microbiome into this space. Interesting, yeah. But nevertheless, iRepertoire, it is one of the OGs in the whole space. So I would say you're there for 2.5 years, but I'm sure you're familiar with their origin story and how they get started. Can you elaborate on this maybe a bit?
Yeah. So Jin Han is one of the original founders, and he's kind of a serial entrepreneur. So he started 4 different companies. 2 of them he has sold off. One of them got sort of rolled into iRepertoire. That's the iQubate automation company. So iRepertoire is very near and dear to his heart, because that is his expertise, is immunology. And yeah, company was founded in 2009. Jin's background, he's multiplex PCR originally for like infectious disease and pathogen detection. And in speaking with him about the origin, he was able to develop these multiplex PCR assays to detect pathogens, but you couldn't really predict what the outcome was going to be, like what's going to happen in this particular individual, this patient. And so that really started the idea, like, oh, we should take this multiplex PCR and we should look at the immune repertoire. And this was right around the days of like Roche 454 sequencing and the beginning of next-gen sequencing. And so, yeah, this was really at the very early days of next-gen sequencing, which is perfectly suited to immune repertoire analysis, because there's just a tremendous amount of diversity there. And how do you capture that? How do you leverage that? And yeah, so that really started this company, very focused on immune repertoire analysis.
Oh yeah, indeed. Yeah. So it is initially just trying to use this PCR technology to, and then leveraging the whole revolution of next generation sequencing, jump on, and use that to profile this whole diversity. Okay, interesting. So yeah, that was back in the day. How old is iRepertoire by now? Like, how long are they operational?
Oh yeah, I think we're maybe 17 years.
Okay,
Yeah.
wow. Okay. Yep. Cool. Yeah. And for those less familiar, indeed, you had this origin, that's the start of the whole company, but I guess products evolve, solutions evolve. What would you say, like today, what is iRepertoire's most famous for, or most unique for? What would you say that iRepertoire's core business is today?
Yeah, they started with single receptor chain repertoire analysis. So that was their ARM PCR, Amplicon Rescue Multiplex PCR, which has been branded as RepSeq. And in 2017, they redeveloped that chemistry, actually completely redeveloped that chemistry, addressing some of the shortcomings of the earlier version. So yeah, one of the challenges with repertoire analysis, when you're co-amplifying all those different V genes together, even with the best primer design, you're going to have some V genes that are slightly over-amplified. So this issue of V bias creeps in. It's a systemic error, so it's there in all libraries. But the newer chemistry, which is dimer-avoided multiplex PCR, it addresses this issue of V bias. It also eliminates any short product formation, so there's no PCR primers that are allowed to remain in the reaction when you get to the PCR step. So it's very specific for amplifying the VDJ. And when they developed that chemistry, the thing that Jin really wanted to do was to make it a complete adaptome. And so being able to amplify TCR, alpha, beta, gamma, delta, and B cell heavy and light chain with all the different isotypes and the sub-isotypes, to have a single molecular assay where you can capture the entire adaptome, that was really his core requirement for that chemistry. And that is far and away the most popular platform that we have right now. It's probably over 70% of our business is the RepSeq Plus platform.
Okay, interesting. Yeah, try to profile everything at once instead of just focusing on a single chain or a single. Okay, cool. Yeah. Okay. what I also feel like as a scientist, you can get information a lot out of papers, but what's living now and what's really on the minds of scientists in biotech and biopharma, for example, is just something you know by talking to them, by talking to customers. I do wonder, like indeed, from your side, as a executive director there, I guess you have a lot of customer conversations. So what is one of these most frequently asked questions by your customers? Like what's one of the maybe misconceptions in that regard, or what do they really, what's the most frequent asked question by your customers?
Yeah, so looking at the market and the customers that we support, they kind of break up into four main application areas. The largest one would be biomarker development and like disease endotyping. So, you know, you have one particular disease that clinically, that maybe there's different subtypes to that disease, but it's not, you can't really distinguish it clinically. And so looking at a molecular method to parse, you know, so lupus would be a good example. So the difference between IDLE and SLE, it's really obvious when you look at the B cell repertoire, but clinically you look at a skin lesion, you can't really tell the difference. So there's a lot of that. I see a lot of that both in the pharma space as well as in the academic space. And then there's always like the vaccine development and yeah, cellular immunotherapy. We're seeing a lot more in that area, monoclonal antibody development. But yeah, biomarker development and disease endotyping is probably the number one application bucket that most of the customer conversations I have fall into. And in terms of misconceptions, I think that probably the biggest one is that V beta alone is sufficient. You know, people are like, oh, I can just look at V beta and I can see whatever I want. It's like, yeah, no, there's actually a lot more going on in the immune repertoire. And it's interesting. I talk to people. The T cell people don't really want to talk about B cell. The B cell people don't really want to talk about T cell. So there does seem to be sort of this chasm between the two. So we are trying to work towards bridging that gap and saying, hey, you know, there's a way to generate a molecular snapshot of everything. But yeah, I think the misconception is that V beta alone is sufficient because it's the high diversity chain. You should look at that one. And what we've been finding is that actually the lower diversity chains can be more informative for understanding and endotyping different complex disease pathophysiology.
Okay, that's interesting. It's like adding a new microscope, like instead of just focusing on this one molecule that changes, you add different chains, different kind of isotypes, being able to maybe stratify better between patients' population. Ah, cool. Interesting. Yeah, but it's also a good segue indeed. Like you mentioned that a lot of people say V beta is good enough, but yeah, this also brings me when I just started in the whole field. T cell sequencing on itself already offers many choices. You can go from DNA versus RNA, single cell versus bulk, sorted versus unsorted populations. I guess you get a lot of customers that are in different phases of the process, like some exactly know what they want to do, others still have to be guided somewhere. How do you guide your customers in making these kind of decisions?
Yeah, I try to start with what their downstream data analysis goals are, because that really informs what starting material we should be looking at, which platform we should be looking at. And that's one of the really unique and differentiating things about iRepertoire. We have, I think it's 11 or 12 different patents related to amplifying and analyzing the immune repertoire. So we have a very deep technology stack that we can bring uniquely to every client project. And so it's not, oh, you should do RNA, or oh, you should do this. It's, well, what is it that you're looking for? What's your hypothesis? What's your study design? What is it that you're trying to extract from this particular study? And then really that shapes the discussion. Yeah, we talk to a lot of people that have had experience working with adaptive, and so they're very focused on genomic DNA. They care a lot about quantitative. We have a quantitative genomic DNA platform, so we're happy to support customers and maintain that continuity. But if it's apparent that their goal is really biomarker development and they want to capture very rare, low frequency antigen specific cells, you don't really see those very well from genomic DNA. So if you think about it, from genomic DNA, you know, there's a lot of genomic DNA mass that goes into the library, but a very small amount of that is the VDJ, let alone a fully spliced VDJ. So you have a lot of partial splicing products that can generate non-functional reads. So we do tend to see more unusable data in genomic DNA as opposed to RNA. And then if you have myeloid cells, non-lymphocytes that are present, those dilute the signal even more. So genomic DNA is not the most sensitive starting analyte. So if the goal is biomarker development, capturing everything, I'll try to, convince people, hey, RNA, it's expressed. There's many thousands of copies. Rare cells we have a much better chance of picking those up. And yeah, the immune repertoire, it has this long-tailed distribution. It's very easy to capture the head of the repertoire distribution. It's very difficult to capture the tail, especially from genomic DNA, because those low-frequency clones, there's one VDJ for that cell, and you're going to sample those stochastically. The other one is single cell. So yeah, 10x genomics is really, really popular, and I think it makes a lot of sense for very broad immunophenotyping, because you can do gene expression analysis. You can look at what's going on with the gene expression on a single cell level. But I think what people don't really think about, and I run into this a lot, is people are very overconfident in the method, and they don't really think in terms of trade-offs. So when you're doing single cell sample preparation, you're losing a significant portion of that sample, especially low frequency rare cells. And so that has a real impact on what you can do with that data. But I think, yeah, there's always this fear of missing out, like, oh, I have to have the pairing information. If I don't have it, then I'm missing something. Well, you are missing something, but it's what you're missing, or the half the repertoire, or a significant portion of that repertoire. It's not even surviving and making it through to the final step in the workflow. So yeah, it's interesting. You talk to a lot of people, and in sales, you learn to ask really good questions. I talk to people that are smarter than me all the time. And so I've learned that I cannot influence people by telling them anything. I have to ask questions and get them to start thinking down maybe the road that I think they need to go down. But not in a manipulative way, more in I genuinely want to help this customer achieve success, for their stated research aims and objectives. And the best way to do that is by asking the right kinds of questions that get them going down that road. And it's not uncommon for us to do a pilot study where we'll co-extract genomic DNA and RNA together.
Ah, really? Okay.
And do a pairwise analysis, because at the end of the day, you have to prove it. You have to show people. It's like, okay, here's genomic DNA and here's RNA. Look at how much more sensitive RNA is. On average, we pick up like 5 to 10 times more clones from RNA than we do from genomic DNA. So there are definitely strengths and weaknesses to each technology, and it's nice to have so many different technologies, and we can bring them all for every client.
Yeah, that's also really nice. You start off with figuring out what exactly they need, what they want, and you have this whole repertoire of solutions available that you can actually use and give them, and even set up pilots to show them like which one, actually solves their problem much better. Yeah, it's interesting. Right. Yeah. Okay, yeah, maybe you've mentioned already. There's a lot of things to be seen. It's easy to capture the head of a repertoire, but sometimes there's also interesting stuff in the tail. So is this something like, I'm thinking about maybe one of the most exciting projects that you've seen being used with your technology. Is this an example where people actually find stuff more in the tail, or what are one of these what would you say is one of the most exciting projects that you've been able to work on?
Yeah, I don't know that I have a favorite one. I mean, we've made a number of case studies where we put things together, like our favorite publications, things we've worked on with different customers. In the last year, probably the most interesting application is cellular immunotherapy. So Innate Bio is another company here in Alabama. And so what they're doing is they're manufacturing a gamma delta T cell therapy, and it's really neat. It's almost like they've made these things Roundup ready. Like one of the modifications they do to the T cell is they make them immune to the chemotherapy. It's like, oh, that's really cool. The project we worked on originally with them was like glioblastoma. So you take the tumor out, and then they modify these gamma delta T cells. They make them immune to the treatment, and then they put them right back into that location, and yeah, the progression-free survival, it's really impressive. So yeah, cellular immunotherapy is interesting, and when we start these conversations, there's a number of companies that we're working with in this space, but usually it's monitoring. You know, they want to look and see which clones are surviving, which ones are expanding. So they're monitoring what's happening with the therapeutic product after it's put into the patient. But the other part that's really interesting is what's the impact on the manufacturing process? You know, pre and post, you know, gene transduction. Are they skewing the repertoire? Are they preferentially enriching for certain clones, or are they putting a complete repertoire into that patient? And so that's been very interesting to see how that's really that level of analysis has informed even the therapeutic manufacturing optimization for cellular immunotherapy. There's a number of like cancer vaccine projects that we've done. So we did a case study. Neon Therapeutics was one. So it's cancer neoantigen vaccine, and it was what was really interesting in that study. So this was for melanoma. They published using our method. They ended up getting acquired by BioNTech, but in that study, so they had these patients, and they gave them a pool of neoantigen peptides, and then for the patients that had a really strong response, they went in and did both bulk repertoire. So they did pre and post looking at the bulk repertoire. And then they used single cell, and the single cell method that we have at iRepertoire is it's not single cell RNA-seq, it's single cell TCR-seq or single cell TCR-seq. So you're labeling antigen specific cells like with a tetramer, and you're sorting them into a 96 well plate, and then we literally do a 3 microliter library prep. So it's all done on a robot, it's all automated. But what they did is they captured the antigen specific receptor pair, and then they cloned it into a Jurkat T cell line, and they re-challenged it with the peptide and measured interferon gamma. So it was a lot of work, but they proved it's like, oh, okay, these patients responded really well to the vaccine. Let's go clone that sequence, put it into a Jurkat cell, re-challenge it, and prove that that was exactly what was going on. So that was really cool. And then there was one other study that we did. So this was the first fully human monoclonal antibody for neutralizing malaria, and we have a case study on our website for this one. But yeah, this was also a multi-dose vaccine, and they wanted to capture the antigen-specific B cells, and so they used a streptavidin molecule, and they put their biotinylated peptide on this fluorescently labeled streptavidin and used that kind of like a tetramer to label the B cells. And the cells were so rare. I think we went through something like 50 mils of blood, and we captured like maybe a dozen B cells, like very, like not very many cells. But they did this for, I think it was 8 different patients in the trial. They cloned the sequence, and then they did the antibody recombinant expression and affinity characterization. And what was interesting there is, all the patients produced antibodies that bound the antigen in the vaccine, but it's denatured. One particular receptor pair was specific not just to the denatured protein, but also to the native protein at all the different sexual stages of the malaria parasite. So really potent in neutralizing, and so that turned into a Nature publication. So
oh wow,
very hard to find those rare cells, but we were really happy to be able to support that project.
Yeah. Now I understand why it's hard to answer this question with just one single project. It all seems like very interesting applications of the technology that you're offering. Okay, cool. Yeah. Maybe going towards the end. So yeah, you've been working at iRepertoire for 2.5 years. How do you see this field actually evolving, like the TCR sequencing field? Do you see any big changes coming up? Like what do you think is going to happen in the next few years?
Yeah, my thought is that people are going to now, once the AI is really helpful. So AI being able to understand the significance of the repertoire, tools like your Detect platform, and being able to annotate the repertoire, what is it that these clones are doing? What are they responding to? And bringing some of these language learning models and different machine learning algorithms to bear for these really complicated data analysis. I think that's going to be really transformative and allow people to look at the adaptome as a new layer of the multi-omic picture. So when I was at Olink, that was a big part of what we were presenting to the market was that proteomics is very important. Like, this is another layer of multi-omics. It's critical. You've got the metabolome, you've got the genome, you got the transcriptome, you've got the proteome. I think the adaptome is next. So with AI, like, now I think we're poised to see some really transformative use in this particular area.
Yeah, that sounds super exciting. Like, indeed, it feels like we're relatively good at reading out the adaptome for now. But like the next stage would be we can get, maybe we understand already, can get some cool insights already. But maybe to really go into the next phase, we might need more of these analysis tools. And you think AI could play a major role in there? Oh, that's interesting, exciting. Yeah. All right. Thank you for being here, Jonathan. I really enjoyed this conversation. Where can people find out more about you and iRepertoire if they want to connect?
Yeah, so the website is iRepertoire.com. And if you want to learn more about what we have to offer, I highly recommend clicking on the Learning Center. So that's my favorite part of the website. That's where you'll find all our posters, all our publications. We have a number of ebooks that we put together that explain, like, the challenges of sampling the immune repertoire, how we address it with our chemistry, sample requirements, a lot of the logistics. And then if you want to talk, yeah, we have one salesperson. You're looking at them. So you contact the company, you'll be talking to me, and I'll be very happy to connect with people. Yeah.
All right. Thank you for joining me, Jonathan, for the very first episode of the Repertoire Room. I really enjoyed it, and I look forward to speaking to you again in the future.
Yeah, thank you for the invitation. Very, very honored to be your first guest.